Today we shall discuss on the next part of the biological classification. We have already studied that according to Whittaker 1969, the living livings are classified into 5 kingdoms. One is Monera, 2 1 Protista, 3rd fungi, 4th plantae, and 5th one Animalia. So, where the Whittaker scientist in 1969 classified the livings into 5 kingdoms, that is called 5 kingdom classification, and according to that the first one is Monera, next one Protista, 3rd one fungi, 4th fungi plantae, and 5th one Animalia. Now we shall discuss each one. Now the first one is Monera. This kingdom contains all prokaryotes like bacteria, mycoplasma, actinomycetes, cyanobacteria. This contains all this one. The features of the prokaryotes, that is cellular, nuclear membrane is absent. So, nucleus is ill-developed. Nucleus is ill-developed, and that is called incipient nucleus. That is called incipient nucleus. And next one, membrane bound organelles like mitochondria, Golgi bodies are absent. Second, DNA is naked, means that is no nuclear membrane is found. That's why these genetic materials are not organized in the nucleus, they are in the scattered forms. Bacteria is the sole member of kingdom Monera. Understand? Now we shall discuss about this bacteria. Bacteria is cosmopolitan. It is means it is found everywhere. Like found everywhere, like hot spring, ocean, sea deep, snowy area, etc. Means that's why it is found more or less all the places. Okay. Means in different conditions it is found. Now the forms of bacteria. It is found in different forms like round shaped, that is called cocci, singular coccus, and the many, that is plural, is cocci. Round shaped, this is rod shaped, called bacilli, singular bacillus. This is spirilla and aspergillum, that is quite separate. And next one is that is comma separate. So in the following it is found. Following the coccus may be, if it is found in double, that is diplococcus. If it is found in chain, that is called streptococcus. If it is found in a cluster, that is staphylococcus. So means if it is found in two, that is diplococcus. If it is found in a chain separate, that is streptococcus. And if it is found in a bunch, in a cluster, that is staphylococcus. Similarly, bacillus is found in two, that is diplobacilli, and in chain, that is streptobacilli, like that. So in the different forms these are found. Now in some bacteria the flagella is also present. Flagella containing bacteria. Flagella is a locomotive organ. For the locomotion it is used. On the basis of flagella, bacteria is divided into two. On the basis of flagella, it is divided into two. One is flagellated, in which flagella is present. One is non-flagellated, in which flagella is absent. Means if flagella is present, that is flagellated, and if it is absent, then it is non-flagellated. The flagellated are also called trichus, and the non-flagellate are called atrichus. So That is, if the flagella is present, that is called trichus. If it is absent, that is known as atrichus. And this flagella is this is whip shaped. This contains three parts. One is basal, second part is hook, and third part is filament. And this is attached with the body of the bacteria. Now the flagella on the the flagellated may be of following types. If the flagella is present only one side and single in number, that is called monotrichous. And if it is found on both ends, that is amphitrichous. And if it is found many in number and in one side, that is known as cephalotrichous. And if the flagella found many in number and on both sides, that is called lophotrichous. And if it is found throughout the body, that is known as peritrichous. So it is of following types on the basis of flagella is present, and in the non-flagellate there is no any types. Next is type on the basis of staining. On the basis of staining means on the basis of coloring. Bacteria is of two types. One is called gram positive and next one is called gram negative. This is due to a staining of that, and this was first discovered by S.C. Gram. On his name, this name was given. First, we take the bacteria and drop crystal violet for 30 to 60 seconds. Now, after that, washed by water, then add iodine, and again finally washed by alcohol or acetone. After that, then examined if the purple color is present, then it is called gram positive, and if it washed away, this is gram negative. So on the presence of this color, there may be gram positive and negative. Now we see why some bacteria take a stain and means that is gram positive, and why some do not, that is gram negative. Now it is the bacteria. In the bacteria, there is a outer thick lining of this is the thick lining of polypeptidoglycan. That is the thick lining. And in some bacteria, there is also polypeptidoglycan. But this is thin lining. In addition to this, in that where is thin lining of peptidoglycan, there is an extra coating of lipopolysaccharide. So in some the thick lining of polypeptidoglycan, and in some thin polypeptidoglycan, and in the thin linings, in addition to that, there is an extra coating that is called lipopolysaccharide. So due to presence of this one, due to presence of lipopolysaccharides, the stain does not go inside, entered the wall, while in that where is no this lipopolysaccharides, the stain entered into the cell wall, that the color is maintained there, and that it is in such condition it is washed away. So they are called gram negative, and they are called gram negative, and they are called gram positive. So due to these reasons, that some take colored and some do not take colored. Now the bacteria, nutrition in bacteria. Bacteria is both autotrophic and heterotrophic. In the autotrophic, there is both photosynthetic and that is chemosynthetic. So all type of nutrients are found in that. On that base, the Monera is divided into Archaebacteria and Eubacteria. These are the, now the first we shall discuss on the Archaebacteria. They survive in most difficult situations. Some found in the salt area, that is halophiles. Halophiles is found in the salty area. Some found in the hot springs, they are called thermoacidophiles, that is some are found in the marshy area, that is methanogen. And this methanogen also found in the gut of some ruminant animals like cows, buffaloes, like that. And so that's why their dung is used for the production of methane, that is biogas. So due to presence of this methanogen in the gut of these animals, like cows animals, their dung is used for the production of methane as biogas. The cell wall of the Archaebacteria, the cell wall of Archaebacteria is made of polysaccharides and protein. Polysaccharides plus protein means the cell wall of the Archaebacteria are made up of polysaccharides and protein. So, and they are semi-rigid in nature. So they protect them in difficult situations. That's why it can survive in the many difficult positions. Now the next one is Eubacteria. This is also called true bacteria. Now true bacteria comes under the Eubacteria. Their cell wall rigid, they are motile if they have flagella. If they have flagella means singular and flagella is present. They are unicellular, colonial, filamentous, found in water, found in fresh water, marine and terrestrials. They contain photosynthetic pigment, so that's they are photosynthetic in nature, means they can make their food by means of photosynthesis. Photosynthetic in nature. Example, cyanobacteria. some eubacteria like nostoc, Anabena, they contain heterocyst and help in the nitrogen fixation. So these are the some photosynthetic bacteria which is found in the eubacteria. Now some chemosynthetic bacteria which comes under the eubacteria. Chemosynthetic bacteria, they oxidize the inorganic substances like nitrites, nitrates, ammonia, and release energy for the formation of ATP. Understand? And it is also important in the recycling of nitrogen, phosphorus, Fe, and sulfur, etc. So they are the chemosynthetic bacteria. They use chemicals for their as their foods, and it oxidizes these followings. Now the heterotrophic bacteria. It is found in abundant amount. It is found in must in nature. It is found in abundant it is found abundant in nature, and they are useful for making curd. It is also useful for making antibiotics. It is also useful for antibiotic. It is helpful in the nitrogen fixation. It is helpful in the nitrogen fixation in leguminous plant, and also caused several diseases. Like typhoid, cholera, etc., tetanus, etc. They are the heterotrophic bacteria.